Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Ram Aiyar. Ram is the CEO of Cambridge, Massachusetts-based Korro Bio. The company is developing medicines that edit RNA instead of DNA, which more people have heard about. Previously, Ram co-founded Corvidia Therapeutics and served on the management team all the way up through its $2.1 billion acquisition by Novo Nordisk. He's had a long and interesting set of experiences in science, large pharma, venture capital, and startup operations. In this conversation, we cover Ram's career journey and how it's led him to this moment when it's possible to develop drugs that can precisely intervene with underlying disease processes at the level of RNA and be redosed conveniently for patients who may or may not be good candidates for a DNA editing treatment of the future. Please join me and Ram Aiyar on The Long Run. Ram Aiyar, welcome to The Long Run.
Thank you for having me, Luke. It's a pleasure to be here.
So, Ram, I have to confess, one of the real reasons I asked you on the show today is that you participated in one of my Kilimanjaro expeditions almost a couple years ago, and we talked some on the trail, but actually not that much. So I'm actually curious to learn more about you and your company. You were just kind of, I don't know, one of these quiet, heads down kind of guys. Like you, you just had a good time and mingled through the group and didn't cause any drama. And you were just a really great participant. And I want to know more about what you do now.
Well, it was a once in a lifetime experience for sure when I agreed to go with you, Luke, and the rest of the crew for Fred Hutch. That was really a great experience. Climbing Kilimanjaro is not something that I had planned or was on my bucket list even 5 years ago. And so to do that with a group that you put together and raising money for the Hutch and spending time with other biotech executives in a very different environment, I think was a pleasure in and of itself. And those friendships that have been made have been let's just say they're sealed right now for a lifetime.
Yeah. Yeah. I love seeing that. And also seeing what people go on to do after being on one of these expeditions. So let's dive in and start from the beginning. Where did you grow up?
Luke, that's such a complicated question. I knew that was coming based on some of your prior discussion. So I've actually grown up in three different continents. So I was born in Bombay, now Mumbai, India. I spent about 10 years there. Then my parents moved to the Middle East, to a country called Bahrain. There was a DoD school there as well. Spent 10 years there, and then came to the US for my graduate work. So have been here for a big chunk of my life, and then spent about 3 years in Europe, doing, being an investment banker, going to B school, also being part of a venture group in Paris for a few years. So where I've
Well,
grown
this
up
is
is
this is really
complicated.
interesting, Ram. I mean, you really are one of these citizens of the world. You've gotten a taste of a lot of different places and experiences, and I want to dig into that a little bit more. So you say you're from Mumbai originally. What did your mom and dad do there?
Yeah, my mom was a college teacher, so she had lost her dad very early in life, and so she had started to work and make a living, when she was 19 or so, and supported both her sister as well as my grandmother. And my dad, who grew up in a small village in South India, ended up coming to Mumbai, working there, was an electrical engineer and was working on ships when he first started as an electrical engineer, and then he moved to the Middle East to continue that electrical engineering journey, eventually ending up in working for an the procurement sector of an oil company.
Oh, so what was it that brought him and your family to Bahrain?
So his job, yeah. So in after I was born, before early in my lifetime, he made a leap of faith to take the risk with my mother to say, hey, I'm gonna go try out this gig in the Middle East for a couple of months. If it works out, I'm staying. If it doesn't, I'm coming back. And he was there for 30 years.
Now, Bahrain, that's a Muslim country. Are you Muslim?
I am not. Bahrain's one of those places that, at least initially, even before the UAE and Dubai got very liberal, was one of the most open places. In fact, there is a British presence there as well as a US base there, and so yeah, they're actually pretty open and liberal. In fact, that was the only place you could get alcohol in the Middle East for an extended period of time. So had no problem. Mostly expats. The island had about 4 million individuals. 2 million of them were expats from various different countries.
So you went there at age 10. Was this a pretty welcoming experience? Was it easy to fit in?
It was very easy to fit in. The reason I say that is because I think from the Indian diaspora, or the Southeast Asian diaspora, a lot of people are already there. There's actually a school, an entire school, K through 12, that only taught the Indian curriculum. So you could basically study in Bahrain and still follow the same curriculum you did in India. Um, and we had temples, we had churches, we had mosques. I mean, it was a very, very diverse place. So growing up, you didn't have any of those challenges of living in a different country. It was very straightforward, except, we had the luxuries of having Coke and Kit Kat and Snickers and all of the Western joys that you could have that we didn't have in India.
And your dad was, he worked in the computer industry, and it sounds like he stayed for a while. This went pretty well. What kinds of things were you interested in as a kid? Were computers part of it?
Did a lot of work on electronics. Used to build a lot of circuits. Legos were always up there, and then started to do a lot of sports. So played basketball, any sport I could get my hand on, but basketball became the sport of choice. And so, given there was a American school and an American base there, my mom ended up teaching there. She taught IB there, and so I would go and play basketball multiple times with folks there. So that's was a good training ground for me.
Okay, given your age, was this like when the Bulls were at their peak and Jordan was was the man?
When the Bulls were at their peak, at least the first three, and then we used to get to, because we lived in the Middle East, and there were a lot of British expats. We also got to see a lot of soccer. So English Premier League was also huge, and so it's during those times that best of both worlds. You got to experience a lot of sports, and Bahrain was a very safe place.
Uh-huh.
So,
And
you know,
what kind of student were you?
oh, that's a tough one. What I would say is that my early days were not very good. I was more hardworking than smart. Over time, I got more interested in biology and math, even though that's eventually is not what I went from a education perspective. Those were two areas that I appreciated and liked. But
Okay,
I
but
was
as like a high school student, you hadn't really figured out what you wanted to do, or you weren't applying yourself, it sounds like.
No, not in the least. I mean, I remember my mom used to tutor IB students, and they'd be home, and I'd come back from school, and I would sort of dabble, but never really super interested in education at that point in time. Though my family is, as Indians, as you can imagine, we're pretty big from an education standpoint, but not really I wouldn't say I was a student at all. I was just maybe coasting is not the right word, but good enough without without needing to do a lot.
Uh-huh. And how did you end up coming to the United States?
So I ended up doing my undergrad in Bombay. I went back. I wanted to get to med school, but med school was very expensive, especially from somebody coming as an expat into India. And so I ended up doing Mumbai University. I did my um, Bachelor of Science in Engineering, Electronics Engineering for 4 years. And then post those 4 years, ended up coming to the US, did my master's and PhD at Drexel University. And at least from a master's standpoint, when I came here, it was to work with a professor called Dr. Moshe Kam. And the goal there was to really understand what now people call neural networks and machine learning and AI. But back then it was statistical pattern recognition and trying to understand and make sense of distributed regions of information and how do you make sense of it. So ended up coming to the US to do my master's in electrical and computer engineering, and joined Drexel.
This sounds pretty hardcore. Like, were you thinking in those days that you were going to be a computer scientist or an academic, or what were you thinking?
So computer science for sure. I mean, I did as part of my undergrad, I ended up doing a signal processing project at the IIT in India, and it was very interesting to use sonars and radars to understand information and see objects. And so when I came to work with Moshe at Drexel, the idea was, how can we use disparate sources of information and really put it all together and make sense of it? Even back then, I think it was very complicated. Neural networks, were identified with Hopfield networks maybe like 30 years before that. And we were trying to make sense of, okay, how can we utilize these black boxes with, what now people call deep learning, but neural networks to see if you can make sense of it. So that's really where I cut my teeth in. I didn't think I would ever be an academic. I wasn't smart enough. I would, it was pretty clear to me that, I was more of a practical person, and needed to have practical experience. And so getting into academia was not in my cards for me.
Okay, but it's still very much in the tech world. You mentioned that, I mean, you became interested in biology at some point. When did that happen?
I was always interested in biology. In fact, the only shining grace in my high school academic career, if you may want to call that, was I taught my school in biology, and my mom used to teach botany. So there was always a link to biology for me. But I got into biology after when I graduated with my master's. I ended up going to Bell Labs. Actually got laid off very early in Bell Labs there, literally within a couple of weeks. And then I came back to Drexel, and my advisor was like, "Why don't you teach?" I used to teach a sophomore class of differential equations, and so he said, "Why don't you teach? You can hang around here and see if you find a project. Would love for you to do a PhD." And at that point in time, I was like, "No way! I'm not spending 3, 4, 5 years in academia trying to identify novel insights." But somewhere along the way came two projects, one from GSK to look at proteomics-based data to understand, can you detect, can you identify, can you predict toxicology in rats based on information that they've generated, high content data. And then the second project I got was from J&J, which was to differentiate three different drugs, all EPO agonists, based on high content data. So 12 color flow cytometry in this case. So if people don't know what flow cytometry is, it's measuring single cell parameters based on fluorescence. And so we would have, call it 100 million cells to look at, and we would have probably around 20 dimensions to look at them in.
Yeah, you can count the cells and get quantitative data.
Exactly.
So what years was this?
So I started Drexel in '99, and so this was 2000 through 2005.
Okay, so early 2000s, the genome was in the news, of course, then, but all these other types of data or the tools really were throwing off large volumes of data, whether that be quantitative cell counting via flow cytometry or proteomics, proteins in large volumes. And suddenly, well, now biology is becoming a little more of an information science. Computers are more useful. It sounds like these things were coming together for you.
It was a perfect storm, right? Because I had folks in my lab that worked on, Affymetrix, had just got their chips out. There was so much data. People didn't know what good looked like because, from a biology standpoint, you didn't really have truth. And so, just understanding what basal level of data was, what is noise versus not, you know, in those early days took a lot of effort to try to figure out, what is real versus what is not. And so some of those challenges still exist depending on the technology. But I think it was very interesting to see that quantitatively, there was so much more information out there that we actually suffered at that point in time. We suffered what is known as the curse of dimensionality. So for each sample that we would get, whether it's from a mouse, whether it's from a human, you could find millions of attributes, but it was just one data point. And so the curse of statistics is that if you have that many attributes for one sample, then you need a lot of samples to make sense. And in biology, you never have enough, whether it's clinical studies, whether it's experiments you're running with mice. So living through that period was very, very interesting because the fundamental algorithms that were developed are still being utilized as we speak.
So those two projects that you cited, one for proteomics and the other in flow cytometry, I mean, were they successful or did they kind of leave you hanging and wanting more?
I would say I got lucky. The project that I worked on with J&J using flow cytometry, actually, we were able to generate a hypothesis. Knowing my curiosity around biology, because it was done with J&J. My mentor at that point in time said, "Why don't you come and run some experiments? We'll hook you up with another research scientist." And the two of you can generate the data. You can dose the mice. You can collect the spleens. You can squish it up, look at the cells all by yourself, and it will be a great learning for you. And so I jumped on that because it's not common that big pharma lets you, an outsider without going through all of the checks and balances to come and run experiments. And so I was lucky enough to be able to do that. And so that project actually became my PhD dissertation,
Had you
where
even held a pipette by this point?
nope. Didn't know what I mean, I did high school biology, but those were glass pipettes that you had to use and draw liquid through sucking on the other end. And so coming and working on a multi-channel pipetter was not something I even looked at.
So somebody rolled the dice on you and gave you a chance.
Many people did, right? So my advisor, Moshe, when I came to Drexel, gave me a chance to work in his lab. My mentor, Peter Bogalski, who's unfortunately no more, he gave me a chance on coming into the lab. And Ren Kapakasal, who's still a friend of mine, runs a flow cytometry CRO. Yeah, he taught me all the basics around how the technology works.
And this didn't scare you? It felt like it suited you to go down this road?
I was a kid in that candy store, Luke. Wanting to come back and have a impact on biology was something that I always wanted to do. And so having this way to, work at a lab, work at a large pharmaceutical company, work on potential drugs, I think was very, very interesting. So much so that some of the work that I did, we actually translated that. Now you're seeing companies like gene editing that are increasing fetal hemoglobin to make way for sickle cell. We had identified some of the differentiators uh, through those experiments that we could activate with some of those compounds, fetal hemoglobin, filed a patent, and we didn't take that any forward. But those were, as a young scientist, if you may want to call it, to have something meaningful I would just say I just got lucky.
Wow, that's really interesting and small world. You might not know, but I was on Kilimanjaro my last trip with one of the first patients who was cured of sickle cell disease with the gene editing that basically turns on his fetal hemoglobin.
Yeah,
um,
no, I do know that. And it is a small world. These things keep coming back over and over again. So, like I said, I think to do that early, I just got lucky. That, finding a drug or having a drug that has a huge impact, you never really know. But to be able to touch it, I think was interesting for me.
okay, so now you weren't done getting educated. You went to MBA school. What were you trying to accomplish there?
So my journey in Janssen, I found many people that took a lot of faith in me at various points in time. Right, so I started when I finished my PhD. I had my mentor because I'd already started work early in 2000s in the lab. So I started working at Janssen pretty early in my life. And so I was there for about 8 years in total. And through that time, started when Peter hired me. Turned out that I got curious enough from a biology perspective. They saw the three alphabets behind my name and said, okay, this guy knows something about biology. But it turns out that, asking dumb questions is sufficient. And I didn't have the baggage of being stupid or otherwise, or feeling that. And so I spent 3 years working on multiple programs within J&J. Then I transitioned to immunology group under clinical biomarkers. I started to work on human samples and using data to distinguish where you can apply certain therapeutics to certain indications. And then I got to a point where I wanted to do more. And so the way that I thought I could do more is apply my science, apply my data science background, and think about where it is I can start investing in. Because by that time, I had touched about 4 drugs or so that eventually got to the market. And so the best way that I knew at that point in time was to take a hard pivot and do a business degree. And in that front, which is in France, Singapore, and Abu Dhabi, provided an opportunity cost that I could have passed on. I got into US schools, both in Philadelphia as well as in New York. But the INSEAD program was a 10-month program, very intense, with an internship for two months, both in Singapore as well as in France. And so my wife, my first daughter, and I decided to take a leap of faith and went.
Okay. That took some guts and to be unafraid to ask dumb questions. You've sort of alluded to this a couple times. Where do you think that comes from? Because I think a lot of people are afraid to do that, and they sometimes specialize maybe to a fault or too much just because it's comfortable in there.
I would say it's a sum total of a lot of experiences I've had, Luke. Part of it, when you're growing up in an environment where, in my home as a child, where you have a teacher as your parent, I think those inherent questions being stimulated was part and parcel for a lot of the conversations we had. The second component is that, in sports, I would usually have a facilitator role, right? So a point guard on basketball, a spiker and volleyball, and vice versa. So it was very inherent for me in terms of supporting and helping. And so a big part of that is asking questions. And then later in life, after I got married, I had the support system with my wife and kids. And so when you have that, I think you sort of end up in a situation where you can afford to be a little bit more confident because you know that you have fallbacks.
hmm You naturally felt a little more like the fox than the hedgehog. I mean, there are ways to go back and forth, and each has their virtues. But it sounds like you found yourself just interested in lots of different things and ambitious to do more.
I would say, leaving India, coming to the US, not having a scholarship, finding a way to support myself, taking a leap of faith into biology, not knowing that. So I think these are all experiences that add up. And if you haven't like failed miserably in any one of them, I think you find that the human spirit can do a lot.
Okay, so you worked at the big company J&J for some early formative experiences. At what point did the light bulb turn on for you that you might want to go work in startups or be an entrepreneur?
Yeah. So went to INSEAD with the goal of getting into investment banking, because back then most folks that got into venture have an investment banking background. And then the idea was to transition to a VC. And so I ended up doing an internship at JP Morgan in London through INSEAD. And then as I came back to France, I ended up getting recruited to Sophie Nova Partners in Paris, investing in both medtech and biotech. And when I was there, I had worked on some early startups, very formative, like just starting physician in the hospital, treated some patients with a compound that he thinks you cannot do this in the US, but you can do it elsewhere. And started to see some huge benefits. And so started to work with him and his founder to set up a company that eventually became something that Sophie Nova funded. So I did two of those. And so got the bug of working in startups, figured that given I had a breadth of experience between how finances work, what is interesting from a technology standpoint, translating an opportunity from what it is in its nascent state to what it could become, having seen drug development being done the right way at J&J, so I could connect the dots in terms of what is reasonable and what's not. So that's what got me excited to work and build startups. And so to this day, even with Korro, that's really been the mission for me in terms of knowing how to connect the dots to bringing medicines forward.
Yeah, that's a really interesting breadth of experiences that you got even before. How old were you at that point?
How old was I? I think I'd left for INSEAD. I was probably top 2 percentile for my class for age. I must have been 32 or so.
Uh-huh.
And then
Well, so you were in, you had some tours of duty in finance and venture capital, and I don't want to spend too much time on those, but then you decided to go all in as an operator. Which was the first one there? Was that Corvidia?
So Corvidia was the one that I jumped in full time. I had helped build three others at the same time that eventually became meaningful companies, either through an exit or through compounds in the clinic. But Corvidia was the one that was the closest because that's one that I jumped in with my remaining three co-founders.
And why
Uh,
did you decide to work on that one?
it's like the story coming together. So it was a anti-IL-6 thesis, and I'd worked on an anti-IL-6 when I was in J&J. It was to stimulate hemoglobin production in the background of inflammation. So I worked on an EPO agonist, so I knew a little bit about the biology. The people around the table were very interesting. It was two cardiologists, me and another drug developer. And it was funded by Sophie Nova as one of the investors when we did our Series A. And so the last component of it, which was new and interesting for me, was it was the thesis for Corvidia was around having a single point mutation in the gene called TEMPR6. And not to get into the weeds in terms of the science, but the pathway controls iron within the body. And so there was a link between a single alphabet change in a gene and being susceptible to IL-6 mediated inflammation. And so when we put that thesis together, it was like everything coming home. And that was very, very easy for me to jump into full time.
Okay. Now, what was your role there? You mentioned the other co-founders were cardio cardiologists. What were you supposed to do?
Yeah, so when Corvidia started, our thesis was around looking for cardiovascular benefit in patients with dialysis. And then over the course of the company, we switched it to not just dialysis patients, but also chronic kidney disease patients not on dialysis. When we started it, I would say that I was the only one outside of the CEO, Michael Davidson, who had some startup experience. And so I was the one that pulled a lot of the business plan, a lot of the science, a lot of the storytelling together in the early days. Obviously, over the course of time, all four of us were very savvy across both the science and business, so much so that all four of us have gone on to become CEOs at different companies. But my role eventually, as I ended up, was Chief Business Officer and Chief Financial Officer at Corvidia.
Okay, so you took on a variety of these traditional business side of the house roles. Um, what's the most important thing you learned there?
So Corvidia is actually very unique, and I don't think a lot of people know the story outside of the final exit to Novo. We were 12 individuals or 14 individuals in the end. We had taken a compound that we in-licensed from MedImmune AstraZeneca that had not filed IND yet, and we had taken it all the way through start a phase three cardiovascular outcome study with greater than 5,000 patients. And because it was an antibody, we also had to manufacture it and get ready to treat 3 million CKD patients. So we had also done manufacturing and set it up in a, how do I say this, in an auto injector. So you can use this at home and inject yourself in a very, very short period of time. Within, I think we raised only about 100 million or so for the entire company. And so, I think three things really stood out for me over the 4-year journey. The first one is um, plan for all outcomes because you just don't know. The second one is even when you don't do anything, you burn money. So maximize time. Experiments that you think you can do tomorrow, think about doing them today. Phone calls that you think you can do tomorrow, do today. That was the second thing. And the third thing is that you need to position yourself for getting lucky. What I mean by that is you just have to take a lot of shots on goal because what happened with us is that a study called Cantos read out and validated our thesis, showing the mortality benefit that we think that we could show. So just operationally, those three things were huge for us for our success. Because even after raising just a little bit of capital, we knew that we'd be very, very successful.
That third item, the readout, was that from some competitor who had success?
So the third item was Novartis ran a clinical outcome study in patients with atherosclerotic cardiovascular disease with an anti-inflammatory called canakinumab, which was an interleukin-1 beta antagonist. And so that study, even though it didn't meet its endpoint, there was a subset analysis that showed that patients with chronic kidney disease, if you block the IL-6 pathway, you're likely to have a 50% benefit in mortality. And
Okay, so that was the fortuitous break that
yeah,
happened in someone else's hands. And then the world looked around and said, "Oh my gosh, these guys have an IL-6 inhibitor and they've put it through all the paces. They've set themselves up and this thing actually might be successful in phase
it,
3."
that's exactly right. I think the one other piece I would add is that when we started the company, we looked at genetics and longitudinal outcomes based on that mutation that I said. And we knew that in the dialysis patients, we could show a 30 to 40% reduction in mortality over a certain period of time. To see that the competitor showed that validated in an interventional study just bolstered, like when you have genetics and you have interventional data, the likelihood of success is just very, very high.
Okay. And of course, the company was acquired by Novo Nordisk for $2.1 billion. I think you said that only 100 million went in. So clearly, the investors, the employees, everybody would be pretty happy with that outcome. And actually, I'm going to put a link in the show notes because you did an interview with Vikas Goyal for Timmerman Report a couple years ago where you kind of walk through the steps that led up to that deal. If you like listening to The Long Run, you will love a subscription to Timmerman Report. This is where you can read my coverage of the most interesting startups in biotech, my weekly Front Points column, and commentary from a rotating cast of contributing writers. Individual subscriptions are available on a monthly, quarterly, or annual basis. Group subscriptions are available at a discount. Go to timmermanreport.com and click on subscribe for more. But okay, so you had this success with this startup, and then what? Do you decide I mean, I guess you could have retired or become a venture capitalist. You could do whatever you want. What did you want to do next?
My goal has always been to get as many medicines as I can to patients. And so two things happened. That transaction happened during COVID. So there's not much we could do from a travel or taking some time from a family perspective. So we were stuck at home. And the second thing that happened is that I had a deluge of information in terms of roles and opportunities that could come my way. And so, there was a point in time where I could have gone in and worked at a venture fund. There was a point where I could have taken another role in another organization. But when I saw Korro, which is where I am right now, and I have been for the last 4 years, I joined in November of 2020. It's the same fundamental thesis that Korro Video was started on that I saw here, which is Korro can make a single alphabet change on RNA rather than on DNA and have an impact on biology. And we do it with a chemically modified version of RNA. So think of inclisiran, but instead of pulling in or co-opting a certain protein that is within every cell called AGO2 or that cuts and silences the gene, we actually co-opt a different enzyme that binds RNA to then redirect it to go and make a single alphabet change from an adenosine to an inosine, so an A to an I. And so that's what got me excited, because I know that if we could have corrected that one change in the CKD patients with the point mutation that I spoke about, we could have impacted a huge number of patients. Instead, we blocked inflammation. And so
Well, and that was a different modality altogether. That was a monoclonal antibody.
that's correct. And
But
so,
okay, so you looked around at different opportunities, as you say, and so you have conversations with. I guess would this have been with Ness in Birmingham, or who was it that you spoke to about Coral?
yeah, so one of the talent recruiters from Atlas had reached out in the context. I then saw an opportunity at Coro. Then I spoke to Ness in Birmingham, and just for the audience, Ness was one of the co-founders in Intellia, one of the three CRISPR-Cas9 companies that started early on with Jennifer Doudna. And so knowing that he had seen the DNA editing space and how you can modify genes, and then working on something that changes RNA, piqued my interest.
Yeah, so this is kind of like the next step. If you can edit DNA, which Ness and others have been working on for a while, um, can you also edit RNA? And I guess I'll just ask you a basic dumb question. Why would anybody want to edit RNA?
Um, So let's think about where DNA editing fits, right? And so when we talk about DNA editing, and there are many flavors of it, right? You can change one alphabet, you can change a certain segment, or you can change the entire gene. And I think that technology is moving so rapidly that you can think about doing all of the above. So where that makes a lot of sense is if you understand the root cause of a disease, and you know that it's that genetic change insertion, deletion, change, substitution of your genetic code that's having an impact on humans, then you can change that component in certain cells and have a huge impact. So that, I think, if you can do for some diseases, makes complete sense. When you talk about more complex diseases, and what I mean by that is chronic, when there is a large number of patients, you want that change to be very specific. You want that change to know that you're only making it in the causal pathway. And you want to make sure that it's on off, right? So when you change DNA, it's either there or it's not there. But chronic diseases in general don't function that way. They are both environmentally focused. You have some environmental factors that drive it. By the time DNA gets made into protein, which is the business end of everything biological, there are lots of changes that occur. So it's possible that even if you have a genetic code, there are some factors in the middle that change outcomes. And so you want to be able to make changes that are not 0 to 1, that are transient in nature, that could be heterogeneous and have a dose related impact. And so that's why changing a compound or a molecule like RNA that's very transient makes a lot of sense. So,
And with DNA, I mean, this is the underlying fundamental code. And if you're trying to edit out a mutation, you're only going to get one chance to do that.
that's right.
Single dose, and you better be right, because if you're wrong, if there are off targets, I mean, it can be catastrophic.
Uh, Absolutely right. But thankfully, at least for some of the indications that people have gone after, that has not been the case. And as somebody that believes and is optimistic about technology, I do think that they will get there, where you can get very specific. However, all technologies are iterative, right? So where an iPhone 16 today was not where the iPhone 1 was.
Well,
So it's just a
right.
question
But
of
back
time.
to your point, though, that there are places where DNA editing is appropriate to give it a shot, like today, here and now with version 1.0 or 2.0, whatever you want to call it. But
Exactly.
there's a whole landscape of other types of diseases where you might want to you might want to intervene with some of these more transient bad actors causing disease, and maybe even more than a single nucleotide that's off.
So let me give you a couple of examples, because I think that examples help a lot in terms of where some of those things make a difference. So um, our lead indication is in alpha-1 antitrypsin deficiency. So it's a disease where the protein is secreted by the liver. It's in response to acute inflammation or pathogens that come into the lung or the skin. And the issue with this disease is that the patients that have ZZ homozygous alleles, or one version of the gene, the protein sort of misfolds, gets polymerized, and gets stuck in the liver and is not available for the rest of the body. Okay. The thing is that it doesn't happen the same way for everybody. So for me, it could get stuck in 90% of the time and really damage my liver. For you, it could get stuck at 50% of the time and is, not that big of a deal until you're 80. And for others who are either smokers or heavy alcohol drinkers, it could have an impact on both. So there's a huge spectrum of this disease. And depending on where you are in life, you could be impacted. So yeah, you can come on board, and once you identify an individual as a ZZ homozygous individual, before they get the disease, you can come and intervene from a DNA perspective. But I don't think our healthcare system is set up to do that in a way that's meaningful and provide benefit down the line. So that's one area where if you have a heterogeneous disease patient population, coming on board with something transient and dosing chronically can provide the same benefit that you can get when you replace the gene, but you can do it in a very transient manner.
I see. So you have this single nucleotide that's off. You switch the A to the I. So that's a very clear biological mechanism driving, in this case alpha 1 antitrypsin, but it manifests phenotypically in different ways.
That's correct. That's correct. And
Okay.
and so that's one example. The other example is, let's say you acutely need to activate a certain pathway. So you don't want it to be permanent. You want to turn on certain signals in your body that can protect cells. You don't want to do that permanently either. You want to do it either in hospital setting or when you have a flare. There are some indications like that that you can think about it. There, something that's transient also makes a lot of sense because you're using it for a moment in time.
Okay.
So
But now, when you started having these conversations about Korro, where was the technology in terms of just our capabilities to even begin to edit RNA? Because these are hard targets, shall we say?
yeah, it was early. I think the team had done a great job in terms of showing. When I joined, the team was able to show that you can deliver a chemically modified RNA. You can deliver it to the liver using what is called as a lipid nanoparticle. So taking a lipid bilayer and encapsulating this chemically modified RNA, deliver it to the river, and we could show that we can edit and we can show impact on protein. So it was very, very early. But one of the things that, again, call it intuition or otherwise, having seen the work come through with Corvidia, knowing that compounds that came out of Alnylam and Ionis have been approved and our drug product is going to be very similar to those, it was clear to me that we could get there. Like we could absolutely get there because the mechanism works, the biology works. Now it's an engineering problem to identify the right composition of these compounds that can get to the right level of editing that we would need and a right level of biological impact. So, uh,
So there's a certain
it,
element of standing on the shoulders of predecessors, both with the making of oligonucleotide compounds and lipid nanoparticle delivery. You're able to you don't have to reinvent those wheels necessarily, but there's more to it. What was the hard part, the key technical challenge that you guys had to address?
so the thing that really helped Alnylam and Ionis was a crystal structure around how their compounds bound the protein and how it actually went on to silence genes. A lot of it was empirical, but there were some key scientific discoveries that happened halfway through their lifetime where they said, aha, this chemical modification makes a huge difference to the potency and the stability of these modified RNAs. And that's what's going to drive improvement in editing. So they started with lipid nanoparticles, and then that was IV. Then they went to sub-Q delivery using what I would call, targeted therapies, which we are using in our second and third programs. And then they went to, trying to find out those targeted therapies for other tissue types. So we're following that playbook in terms of where the technology is from a generation one and moving along to improve potency. So delivery was an important component, and the second component was just identifying the chemical modifications that you would make on each of these nucleotides or alphabets to make them both potent as well as stable, which is different than what Alnylam and Ionis and other companies that are there from a gene silencing standpoint are focused on.
Yeah, you're not gene silencing, you are RNA editing. Um, Now, but you need a way to get to the precise location where you want to make this edit. Um, and that requires a guide RNA, right?
That's correct. And so the guide RNA in this case is actually our drug product at the end. So the way that we can design and target the guide is specifically identifying the location or the adenosine that we want to change. It is so precise that even if there is a adenosine close by, it doesn't really edit the adjacent one. So it's a single alphabet change, really. And so the designs that we have are very, very specific. So there's no off-target consideration. And then the next step, as you alluded to, is just delivering it to the right cell type.
Uh-huh. Uh-huh. This sounds like a big step because I mean, I think that the literature is full of people who've said like this mechanism is pretty promiscuous. There's lots of editing going on all over the place in different cell types, and we want to be more specific.
No, that's right. And so, and again, this is where we stand on the shoulders of our predecessors in terms of having delivery tools that we can test out across different options and seeing, which one fits best for that indication of choice. I think the one other thing I would just want to say for your listeners is when we started and when I joined, after I joined with the board, our focus has been because most drugs end up silencing, suppressing a pathway, or reducing a protein. Our goal has always been how do you stimulate or activate or repair a certain pathway, which is fundamentally different than where a lot of gene editing technologies are or RNA silencing technologies are. And so this is another genetic, call it a toolkit in our toolbox to be able to activate a pathway in certain diseases where you can't do it in any other way.
Yep, yep. And of course, you're not the only ones in this area. I know this fall, Wave Life Sciences produced some early phase 1 data with their program for alpha-1 antitrypsin. What what was interesting about that that data readout for you and the whole field?
It's a high tide lifts up all boats, right? So I think the big question that we had, even though we knew this mechanism is conserved, it's very similar to RNA silencing. You don't know until you go to humans and see a certain mechanism work and then take a breath of relief or joy. So when we did see the Wave Life Sciences limited release, the thing that really caught our eye was these patients with alpha-1 deficiency don't have the normal version of the protein, and then post treatment you see some version of the normal protein. That's a big deal. So not only did the edit change the alphabet from a A to I and eventually a G, but the protein that was produced was also real.
And
That's
they had none of it. They went from zero protein production to something. And now,
that's exactly right.
was that something enough to modify the disease?
We don't think that what they showed with the single dose is enough. I think we'll have to wait and see how that evolves over time, because in a multi-dose setting, we'll see it was also, as they had said, their first dose, so they could go up. And that's where we hope to show in the second half of next year with our clinical study. We're already in the clinic. We'll dose our first participant in Q1 of next year, and we hope to demonstrate that, we can generate that M protein as well to levels that are therapeutically meaningful. So don't know where their data stands or when they will release it, but we hope to show that it's possible with this technology. We hope to show that we can have some profound benefit for this patient population.
But this is a phase 1 study, and so they're going to test a variety of doses, as you will too, and dose intervals. Because this can be redosed, which is an important feature for the ultimate product profile. Well, what are you? Can you just talk a little bit about what you're trying to develop in terms of your ideal product profile for alpha-1?
So for Alpha-1, the first generation product that we have in mind is once a month IV infusion to get to protein levels that are at the lower range of normal or as close to normal as possible in this patient population. Alpha-1 is for folks that most people won't know, it's the fifth most concentrated protein in blood at basal level. And so you need an editing that is north of 50% on an active basis to enable protein levels that are as close to normal as possible. And so we hope to show that in the second half of next year. We believe that an IV infusion for a first generation product once a month when we can get protein levels that high is going to be very, very meaningful for this patient population. There's a lot of the good news is that you now have multiple shots on goal for patients that have this disease that didn't have anything before. That's
And
huge.
you think 50% of normal protein production is enough to effectively treat the disease or bring people back to what we call normal?
I would the genetics, remember the Corvidia story that I said in terms of knowing the genetics and looking at longitudinal outcomes. The genetics says that if you have a single allele over the lifetime of the disease, you will have very little lung manifestation and you'll have some liver manifestation over the lifetime. And so that's why we're trying to shoot to be above that 50% range so that we're as close to normal as possible. This is where genetics is awesome, right? You can make some of these correlations, and nature has done that experiment for you.
Interesting. Now, I noticed further back in your pipeline, I don't have a lot of time to ask you about this, but you have programs for, say, for Parkinson's and ALS. What makes you think that your platform might be useful for those diseases?
So let's take ALS as an example. There is a lot of evidence from a genetic standpoint that in sporadic ALS, this protein called TDP43 is dysregulated, and it's dysregulated because it starts to, much like alpha one, it starts to polymerize, starts to form intracellularly within neurons, and then it causes cell death. And because it gets polymerized, it doesn't do its signaling function that it's supposed to do. And we've been able to demonstrate that using a single edit, just one alphabet change, we're able to disaggregate and prevent that aggregation and also continue signaling. So you can't do that with any other technology. Do I know that it's going to work in this patient population? I think all the evidence suggests that we could, but we have to enter human clinical studies and actually demonstrate that. So as of today, it's a very exciting program. It's for me, I think it's a hope that we have for this patient population. We know we can deliver oligonucleotides to a motor neuron in the spinal cord and certain parts of the deep brain. If we can demonstrate that, I think it would be a very, very meaningful medicine for this patient population.
Long term, Ram, last thing I want to ask you, where do you think RNA editing could someday fit into the great medical toolbox of the future? How big of a modality do you think it might be?
So think about it this way. Let's assume you have somewhere between 5 and 6 alphabets in RNA. A is one of them. So somewhere between 20 to 25% of your entire transcriptome is made of adenosines. And so in some instances, those adenosines are very meaningful and you can modify them to have an impact on biology. And so as we think about it at Korro, our goal is, where are those locations? Where can we look at genetics and get inspired? Where can we look at proteins and look at it and get inspired and find a way to activate these biological pathways? In my mind, I think delivery is the only thing that limits us in terms of this toolkit. And over the next decade or so, you'll see more and more of these medicines come together. Just remember, I mean, as you said in your last conversation, there are somewhere between 5 and 7 oligonucleotide therapies already approved. It's just the start.
Yeah. Yeah. And delivery, I think you're referencing the conversation with Kevin Fitzgerald of Alnylam. Delivery was the key challenge for many years there. And it was harder than a lot of people anticipated. But even if that's the case here, you know, it certainly, I mean, it builds on the track record of others, and the ability to interrogate cells and see, if your oligos are going where they're supposed to and having the desired effect. I mean, it's never been better. Things are moving faster. Um,
Exactly.
do you think we're going to have multiple therapies that edit RNA 10 years from now?
Absolutely. No doubt about it. I'm hoping sooner. And I'm hoping that given there is multiple companies working in this space, I think it will lift all boats. So I'm very, very hopeful that we'll have multiple therapies coming together. And just to give you a sense, all the public companies that exist, we're all outside of Alpha-1, we're all working on different things. So it should give you a sense of how broad the applicability of this technology can be.
Yeah. Yeah. Well, I think a lot of doctors and patients would agree that they like disease modifying therapies that can restore them to normal. And, if you can take them on a, semi infrequent interval, that's pretty nice to have too, whether that's monthly or quarterly or even less.
Yeah.
But they're not being asked to make an absolute all or nothing decision like they are in certain cases with gene editing.
Yep, exactly. So there's a lot more flexibility. And like I said, I think I am very hopeful. 4 years ago, when I came here, my goal was, I'd had a chance to talk to John Marignori and before I started, and a couple of other platform companies like Praveen Tipernini and others. And I was like, platform companies are a way where you can make multiple drugs, and I'm hopeful that that Korro is one of them to be able to come out on top and make meaningful drugs for medicines for patients. So fingers crossed. It's a long road.
Really interesting, Ram. I look forward to asking you about it our next time on the trail together. Thanks for joining me on the long run.
Looking forward to it. Thank you for having me, Luke.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.